Today we are going to learn about DNA microarrays, also known as DNA chips. A DNA microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. DNA microarrays are basically microscope slides or chips that are printed with thousands of tiny spots in defined positions, with each spot containing a known DNA sequence or gene. There are many applications for DNA microarray technology. When they were first introduced in the 1990s, they were used primarily as a research tool, and they are still used in that way today. However, even though it is a relatively new technology, DNA microarrays have many more applications now. Here are just some of them. DNA microarray technology is used in biomedical research. Microarray technology is used to identify emerging strains of pathogens. For example, it was actually used to identify the SARS virus. It is used to identify complex mutations in genes that cause human disease, as well as in forensic analysis and genotyping. It is now being used to screen seedlings and the molecular breeding of crops for our food supply. DNA microarray technology is used in clinical diagnostics. Microarrays can measure predisposition to certain diseases. For example, microarrays are playing a greater role in understanding cancer and in creating targeted therapies, especially for breast cancer. DNA microarray technology is used for the pharmaceutical industry. It can be used to identify drug candidates and also to determine which drugs might be best prescribed for particular individuals based on their body chemistry. Multi-stranded DNA and RNA microarrays can be used to identify new drugs that bind to these multi-stranded nucleic acid sequences, which can lead to the discovery of drugs that actually inhibit gene expression. Microarrays play a role in prenatal genetic testing and are used for autism, intellectual disabilities, and developmental delays. So DNA microarrays have a variety of useful applications. They can be used in different ways and answer different types of questions. For example, one scientist could compare what happens to yeast genes when they are grown in aerobic versus anaerobic conditions. Another scientist could explore how cancer cells are different from normal cells. In the simplest terms, however, DNA microarrays allow a scientist to determine which genes are expressed by a cell or tissue. Let's explore a little more closely how DNA microarray analysis actually works. Here are the basic steps in a DNA microarray analysis. First, a control sample and an experimental sample are collected. Just as an example, the control sample could be normal cell tissue and an experimental sample could be tumor tissue or cancer. Second, mRNA is extracted from both samples and then transcribed into complementary cDNA using reverse transcription. Each sample is labeled with fluorescent probe of a different color. Typically, the experimental cDNA sample is labeled with a red fluorescent dye, and the control cDNA sample is labeled with green fluorescent dye. Next, the two samples are mixed together in a single tube and added to the microarray. DNA microarrays can contain 60,000 or more different DNA sequences attached in microscopic spots on the slide. cDNAs are then allowed to bind to any complementary DNA sequence or oligonucleotides that they can find on the microarray slide. When the cDNA molecules bind to the DNA probes on the slide or chip, it is called hybridization. The slides are then put into a special scanner that is made up of lasers, a microscope, and a camera. The slides are scanned for fluorescent emissions at selected wavelengths in order to generate a high-resolution digital image. The color of the fluorescent shows where the binding has occurred. Everywhere we see green dots, that particular gene is expressed in the tissue labeled green. Similarly, everywhere we see red, that particular gene is expressed in the tissue labeled red. If there is equal expression in the tissue samples, then the spot appears yellow. If we see little to no coloring, the gene is not expressed in either tissue. Finally, the fluorescent intensities shown in the digital image are quantified and analyzed using microarray analysis software. The data gathered through microarrays can be analyzed to create gene expression profiles. DNA microarrays, or DNA chips, are revolutionizing the way scientists study gene expression and have led to many important advances. However, like in all areas of genetic science, there are certain ethical, legal, and social issues that must be considered. Microarrays allow mass screening for a wide spectrum of genetic traits. It generates a lot of information. Ironically, this is one of the biggest strengths, but also poses one of the biggest problems. Because microarrays generate such a large volume of information, it makes it very difficult to determine what legal informed consent looks like for people involved in these tests. It also raises privacy concerns because so much information is revealed through this process. Microarrays can detect predispositions to certain diseases. This is a great scientific breakthrough which may help prolong life for many people. However, learning information of this kind can also lead to significant psychological distress and may cause family conflicts. I mean, who really wants to find out they are most likely going to get a form of liver cancer in the future that is associated with a poor prognosis? Further, we might not know enough yet to determine if a particular trait is abnormal or not. People might be subjected to unnecessary treatments or worry unnecessarily. Furthermore, if an individual learns that they are not at risk for a certain disease like liver cancer, they might become complacent or take risks with their health, for example by drinking too much. Microarrays increase our understanding of the genetic causes of diseases including cancer. They also help us to develop targeted drugs and other therapies. However, the high cost of this technique might cause some issues because it's not accessible to everyone. Many people likely do not know that this kind of testing even exists. In genetic testing, microarrays are used in testing for autism, developmental delays, and intellectual disabilities, which might be useful information for parents. Because they test for such a wide range of traits, microarrays also generate a lot of additional non-medical information, for example, traits like hair color. However, the danger in these examples is in how the information is understood and used. It might play into the eugenics idea that social problems can be solved by creating more perfect human beings through genetics and heredity. It also might play into discrimination and scientific racism and adversely affect people of color, people with disabilities, and LGBTQ individuals. People might choose to terminate pregnancies on the basis of information obtained through microarray analysis or to genetically engineer non-health related traits that they want. Overall, microarrays are a powerful and valuable tool to analyze gene expression. It should continue to be used, however, it is important to proceed cautiously with this technology. Different people have different ethics and morals and might use the information in different ways. This field of molecular biology clearly illustrates how important it is for scientists and researchers to work toward establishing agreed upon ethical and legal standards. Thank you for watching. I hope you enjoyed learning about DNA microarrays today.